Hi, my name is Adam. I am a top scorer and an MCAT specialist here at Shemmassian Academic Consulting, and today I'm going to be walking you guys through an MCAT biology or biochemistry passage. So let's dive into it. So first of all, where is this in the MCAT? MCAT stands for Medical College Admissions Test. It's used for a lot of MD and DO schools in both the US and Canada, and a few other professional schools as well as a marker for admission. So doing as well as you can on this exam will help you be more likely to get admitted to the school of your choice in one of those programs. The biology and biochemistry passage is the third section, right? So there's 4 sections. First section is chemistry and physics. Second section is called CARS, which stands for critical analysis and reasoning section. Then we have a lunch break. Then that third section is biology and biochemistry, which is where the passage we're going through today is from. And then the final section, the 4th section, is psychology and sociology. So when we're in this section, you get 95 minutes for the biology and biochemistry section, and you have 10 passage and 15 questions that are not based on any passage. So a typical timing strategy, kind of to start out with, is to do those first 15 questions that are not based on a passage in the first 15 minutes of the section, right? So you kind of skip through to those. So then you have 80 minutes left to do the 10 passages. Pretty easy to break that down. It's just 8 minutes per passage. So a passage like this, you want to be able to get done in about 8 minutes or less. Obviously today we're going to be taking a little bit longer than 8 minutes because I'm going to be breaking it all down in my thought process as we go through the passage and the questions, which will take considerably longer than 8 minutes. But while you're practicing on your own, or if you want to do this passage before I break through it, set a timer for yourself, see if you can finish it in less than 8 minutes. That's going to be your goal on test day. Okay, so without further ado, let's get into it here. So let's start with the passage. One typical strategy is to just read through the passage, highlight what you kind of deem would be important or you might need to answer the questions, and then you go and answer the questions from there. So we're going to start with that strategy, and let's go here. So MCAT biology practice passage, let's dive into it. So post-operative acute kidney injury, AKI, we're going to highlight that. Oop, that's not the highlighting tool. So let's erase that. And let's grab the highlighter. Okay, let's dive into this here. Post-operative acute kidney injury, AKI. Okay, so that's an acronym, so let's highlight that. It's good to highlight the acronyms in case you see them show up again later. You can know exactly what they stand for. There's a severe complication after liver transplantation, LT, as its deterioration and magnification can lead to an increase in mortality, right? So remember the difference between morbidity and mortality. Morbidity is like how much sickness we have. Mortality is death, right? So eventually will lead to death. Connexin 43, CX43. Okay, we should recognize that word as that is associated with one of our types of junctions. Mediates direct transmission of intracellular signals between neighboring cells, always considered to be the potent biological basis of organ damage, deterioration, and magnification. So this connexin that causes transmission of things is considered, always needs to be considered for damage, right? So I'm going to just why they mentioned that there. Okay, researchers obtained donor liver tissues of patients in order to explore the effects of CX43 on organ damage. Okay, so we're starting our study now. Reperfusion is a biological process used to refill blood in AKI. Donor livers displayed more, okay, so let's highlight this because now we're getting some details. They displayed more severe lobular distortion with necrosis, apparent edema, hemorrhage, and neutrophil infiltration after reperfusion compared with donor liver tissues obtained before reperfusion or normal liver tissue. Okay, so a lot of bad things. I'm not going to highlight all of them, but I'm going to take note that this is kind of showing that after reperfusion is worse than the donor tissues before reperfusion, right? So that's like once they put it into the body, then they reperfuse it, and then we have all all these issues. If we need to refer back to that list, we can later. Or normal liver tissue from patients with hepatic hemangioma. After reperfusion, CX43 mRNA and protein levels were determined as shown in figure 1. Okay, so that's important to note. We have to want to highlight what we're looking at here. We're looking at CX43, which is connexin, which is a protein, mRNA, which would be coding for the protein, and then the protein itself in figure 1. Okay, so let's look at figure 1 here. So we got a little protein graph here, right? So B-actin is showing 3 lines here, and then CX43 only shows 1 line here at 43 kilodaltons. That's kind of important to note. Let's look over here at the X and Y axis of the actual graph, right? So this is mRNA units in normalized to normal liver tissue. Okay, so that would mean like our normal liver tissue is our normal level, which is usually set at 1, which looks like that's about where it is. And then we're looking at whether these other ones are different or not. So then in our donor liver before reperfusion, we are significantly higher, right? Notice this little asterisk. That means that we are significantly different. It's hard to tell with that error bar if it overlaps, which is why it's very helpful when they give us that asterisk. So before reperfusion, it is higher, right? Higher mRNA levels. And then after reperfusion, it's significantly higher, right? So we see an asterisk and a pound symbol, which would mean that it's not only different to the normal, but it's also different from before reperfusion. And then the protein levels as well, noticing that B-actin, so these lines would line up as well with these 3 types, right? The normal liver tissue, the before reperfusion, and the after reperfusion. And it's important to note, right, B-actin is present in all of them, but the connexin protein is only present in the after reperfusion in significant levels, right? You can see that line is obvious, while the other 2 are maybe trace lines, but it's pretty difficult to see. This one's significantly different. All right, let's go on from there. Autologous orthotopic liver transplantation, AOLT. Okay, so again, with a lot of bio or biochem passages, even chem phys passages, we might not understand every word that's in it. So it's okay, just keep moving on. That's not something they're probably going to be asking us about, right? If you're familiar with our MCAT topics and what is likely to be tested / what isn't, a lot of these things are probably a little bit past the point of the MCAT, which we don't have to worry too much about. We're going to highlight this in case they use these words again, though, just to look at it later. Were built by researchers in order to further study AKI. Remember, we knew what that was from the beginning. In case we forgot, AKA is acute kidney injury. Researchers hypothesized that the change in CX43 expression might play an important role in AKA following this type of transplantation, right? So transplanting of the liver. Researchers used heptanol, a well-known inhibitor of CX43, without hepatotoxicity, right? So not going to be toxic to our liver, right? Hepato meaning liver, toxicity meaning toxic, right? So not damaging our liver, to alter the function of junctions composed of CX43, right? So we can go back and look at what type that was if we do need to. Heptanol had no effects on expression or CX43 expression in kidneys or liver, so no effects. So they know it's a well-known inhibitor, but it had no effects on the kidneys or the livers in terms of expression. All right. Heptanol or a sham were injected intravenously into the rats, and the pathological score of kidney cells was determined to analyze disease progression, right? So pathological score, right? We should be somewhat familiar with what that means, but basically the higher your pathological score, the worse things are, right? That's why they're using it for disease progression, right? Because diseases, when we say disease progression, we mean like how bad is it getting? So the higher the pathological score, the worse off that particular type of thing is. Right here we're talking about kidney cells. All right, so let's take a look at this graph here. We got pathological score on the y-axis, and then on the x we have, they're all done 8 hours after reperfusion, right? So they all have the same different time and thing that they've done to it, which is reperfusion. And then we have our 4 different types. We got sham, which looks like it's about 0.5, and then we got heptanol and sham, and it's the same. And then we have liver transplantation, right? And we have a significant difference for the liver transplantation. And then we have heptanol and the liver transplantation. And so we notice that the liver transplantation, the AOLT, is still there, right? Because we're still significantly worse than the sham or just the heptanol. But it looks like the heptanol helped, right? Because we see the star and the pound sign, which means that it brought it down from the damage that we had, so we have less damage. So it seemed to have helped, right? So this is just kind of showing heptanol seems to do its job. Seems to help with the liver transplantation and causing less liver damage, which was the goal. Researchers also believe that the RIP1, right, so that's a new thing, may be involved in function of that CX43, and they confirmed the interaction of the proteins through co-immunoprecipitation, right? So they confirmed it. And then RIP1 is a typical marker of necroptosis, right? So we should be familiar a little bit with this, but generally we can talk about it as some type of death. Okay, cool. So now that we finished that passage, before we go on to the questions, let's talk briefly from our ShemmassianConsulting.com MCAT content guides, a little bit about how drugs work, right? So we talked about heptanol, and we used heptanol to decrease our liver damage, right? And so we're talking about what types of drugs are we going to be tested on, or how they work, right? What's the typical mechanism of action? And a good topic to study for that would be the cell membrane itself, right? Because we're going to have a whole bunch of different ways that we can make changes either in the cell or on the surface of the cell and stuff like that. So this is what a membrane looks like, right? This is a phospholipid bilayer, right? And it's going to be typically organized like this, right? Where we have these heads and then we have these tails, right? And they're going to be organized in that way because these heads are going to be typically what, right? So they're facing what would be water up here, which means we'd be hydrophilic, right? And then these tails together would be hydrophobic, which means what we're looking for, if something wants to be able to travel all the way across, it needs to be both small and hydrophobic, right? Because if it's hydrophilic, it's going to get stuck here, but if it's hydrophobic, it can go all the way through, right? So, and a transmembrane protein can help because it also will have those different components, right? This would be a hydrophilic component. This would be a hydrophobic component, right? Because it can coexist with these middle parts here, and this would be, again, a hydrophilic component. We're going to have a whole bunch of different components, right? So if we're going to be needing to make changes on, say, the nucleus of the cell, if this is the intracellular side and this is the extracellular side, if we want to make a change in the nucleus or even in the cytoplasm, we need to have a type of drug that would be significantly small and hydrophobic, right? Because then if we have those two things together, we can cross that membrane without any problem, and then we can enact changes either in the cytoplasm or in the nucleus or both, right? So if we're thinking we want to impact translation or transcription in any way, we can do that. There are lots of other ways to make changes, but that's just one of them. You can check out some of our other options there on our other content guides as well, but I wanted to mention that briefly before going into these questions, because a lot of times when we're seeing a particular drug that's used, we want to be thinking about how it could be going across and how it could be making a change. Okay, so let's start with question number 1 here. All right, so what type of junction does CX43 most likely form? Right, so we got to think about all the different types of junctions. There's a few of them listed here, right? Gap junction, tight junction, desmosome, hemidesmosome, right? We should be familiar a little bit with how these different types of junctions work and when we're going to be using those types of junctions. So let's go back and review what CX43 is from the passage, right? And it was defined as connexin 43, right? And that would be a gap junction type, right? Because gap junctions use connexins. Our tight junctions are not going to be using connexins at all. Desmosomes, right? We want to think about, right? Our desmosomes have a little bit more transferability. We want to think about actin, and we're going to have our intermediate filaments for desmosomes. And then hemidesmosomes are basically the same thing as desmosomes, except for instead of between two cells, it's going to be between a cell and that little basement membrane that's holding that cell onto it. So yeah, these are almost identical, so we could have ruled them out. The main difference between the hemidesmosome and the desmosome is what type of function it's doing. And then, right, tight junctions don't use connexins, but gap junctions do. So that would be number 1 there. All right, number 2, this is a little bit of a bigger question. Let's zoom out. All right, a researcher discovers a new CX43 inhibitor made of glycerol backbone and 3 saturated fatty acid tails. Which of the following describes the path by which the inhibitor enters the cells, right? So we should be familiar with what glycerol looks like, and also we should be familiar with these terms, right? Saturated and fatty acids, right? So that's kind of a little similar to what we're talking about here, right? These phospholipids would have a phosphate, and then these look like these may be slightly unsaturated, but they could also be saturated. It's hard to tell from this image. But it's going to look something similar to that, right? A glycerol backbone would be, right, 3 carbons in a row like this, carbon, 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 bonded to each other with oxygens together that would have been just glycerol before, which would all be just OHs, but then with the fatty acid tails, they would become something that looks a little more like this. Excuse my bad drawing here, but it would look like that, and then we'd have saturated tails coming off, something like that, right? And remember, saturated just means that we don't have any double bonds or no like kinks that's going to make us kind of like messed up a little bit. That would be what unsaturated would look like. Okay, so that's what we're looking at. How are they going to enter the cell, right? That's kind of what we were talking about there. Right, so the inhibitor recognized by a specific receptor, right? If we're going to be looking at a specific receptor, that would be something that would be most likely largely hydrophilic, right? And these tails are largely hydrophobic, so unlikely. Inhibitor is taken up by a non-specific receptor. Again, kind of similar type idea, probably more hydrophilic. This is probably on the surface of the cell, right? So not a great answer either. The inhibitor diffuses through the cell membrane. That seems likely because these are our hydrophobic tails, so that one seems pretty good. And then the inhibitor passes through the cell membrane via a channel, right? So why would we need a channel? Like either we're charged either positively or negatively, or we're significantly large that we're not going to be able to go across. We're just glycerol and 3 saturated fatty acid tails, so we're still relatively small. So best answer is going to be C here, because we're significantly hydrophobic and relatively small, so we can probably just diffuse right across and make a change. All right, let's look at number 3 here. Researchers decide to interrupt the CX43-RIP1 interaction in the AOLT rat model, right? So they're trying to confuse us here. They're like, let's use as many acronyms as we can. And when it comes to MCAT biology questions, we want to look out for this. This is very common, and they're just trying to make a question that's relatively simple feel more complicated, right? So if we kind of break this down into a much simpler verbiage, right? The CX43-RIP1 interaction in the AOLT rat model they're just describing what happened in the very, very end of the passage down here in the bottom, where RIP1 was a marker of necropoptosis or death, right? And we knew that CX43 was also associated because of up here earlier in the very first pair, CX43 is associated with damage. So it's just RIP1 is just part of that, right? And then so in liver transplantation model, they're trying to kind of do the same thing that they did in the passage. They're trying to break this up, decrease our amount of damage in our liver transplants using a small molecule, or without using a small molecule inhibitor, right? So they don't want to use what we used in the passage. So we want to use some other type of thing. So which of the following will most likely disrupt the interaction? All right, so we got siRNA for RIP1, right? So siRNAs, right, are going to bind to the mRNA for some type of protein and stop that particular translation, right? So because mRNA is what is transcribed, which then would travel to our ribosomes to cause translation. So this is a decent answer. So we're going to keep this one around, A, for now, because that seems reasonable. B, radiation against heptanol. Remember, heptanol was another drug that we tried to use that didn't seem to work on kidneys or livers, but when it came to liver transplantation, it helped, right? So, not a great answer, because radiation against it, we knew heptanol was kind of helpful. So if we did radiation against heptanol, if we put it in, would probably just bring us back to increased damage again. So bad answer. Let's take B out. C, acetyltransferase specific for beta-actin, right? So remember, actin is going to be one of our things involved in one of our junctions. If we put in an acetyltransferase specific for that, remember, so if we are thinking about transcription, and acetyltransferase is going to increase transcription, right? Think acetyl sync activation, right? So we're going to increase our amount of transcription. It's going to be involved with our DNA and our chromosomes and unwinding them. So that'd be great, but this is not really specific to what we were talking about in the passage. We're trying to impact the CX43-RIP1 interaction and beta-actin. And again, we can look at figure 1 as well, where they measured beta-actin. It's not really any difference, so it's unlikely to make a big change there. And then a plasmid producing CX43, remember, CX43 is associated with damage. So if we add more CX43, we're going to get more damage. We don't want that. We want to interrupt it. So this is the wrong direction completely. So B, C, and D are all bad answers. A, when we first looked at it, it seemed to make sense. We're going to go with it. It's probably our best answer here. All right, let's look at number 4 then. Okay, so question number 4 here. Which conclusion about CX43 regulation is best supported by figure 1? Okay, so we want to be really careful with questions like this throughout the whole MCAT, but particularly in MCAT biology and biochemistry section, right? Because they'll throw in this kicker here, right? And they'll say figure 1. They might have some statements in here that are supported within the passage, or are even just true statements from the passage or just from our general knowledge or our outside knowledge, but we need to be careful to only choose the one that's true by figure 1, right? So this is very important to pay attention to when you see this in a question. So yeah, let's break these all down here. So A, all expression control occurs at the transcriptional level, right? So if we go back to figure 1 and evaluate this, right? So we got mRNA and we have protein here, right? So we see significant increases in mRNA both before reperfusion and after, and they're significant from each other. And then we also see this significant change in protein levels. So because we see a significant change in both, it could be transcriptional, right? Because that would be creation of mRNA. It could also be translational, which would be this increase in protein from the after reperfusion. So we could actually have control at both levels. So A, where it says all expression control occurs at the transcription, right? That we want to be careful with extreme words like that, but also it could be either transcriptional or translational. If it was all transcriptional, we would see an increase in mRNA and then only a resultant increase in protein, but we don't see that, right? Because we see the increase in mRNA here for the before reperfusion, and we don't yet see, right, if I highlight this one right here, we don't yet see an increase in protein following that increase in mRNA, which means there's probably more than one control point. So it wouldn't all be transcriptional. It's probably transcriptional and translational, which, look at B here, all expression control occurs at the translational. We know that that's definitely not true as well because we do have a change in the transcriptional level. So both A and B are wrong because of that. So let's look at C versus D. C, donor livers have higher levels of CX43 after reperfusion when compared to normal liver tissue, right? So CX43 compared to normal. And remember, CX43 is a protein, so we want to look at the protein levels, which would be down here, right? After reperfusion, we have a line, and then before we have nothing. So that would be considered to be different. So donor livers do have higher levels. So C is true, and it's supported by figure 1. So we're leaning towards that right now. Let's look at D here. Normal livers treated with heptanol have lower levels of CX43 compared to donor livers treated with heptanol. That may be true, but we don't need to evaluate it, right? Because if we look at the heptanol, right, that's figure 2. So we don't really need to evaluate it because the question's only asking about figure 1. So we can rule out D just because we're talking about heptanol. So good. We don't have to worry about it. We can go back to the one that we knew was correct, which was C. So C is going to be our correct answer there. Okay, so thanks for joining me on this passage breakdown. I hope you got them all correct as well. If you have a second, if you could go into the description and click the little link there to get a MCAT question of the day sent to your email inbox, that'd be great. Also go out and check our MCAT guides out at shemmassianconsulting.com/MCAT, and you can get access to a lot of free content guides. So yeah, I hope that was helpful for you guys, and happy studying to all y'all.